Okay, in this video lecture, we are starting chapter 13, which is titled DNA. And this is a very, very short chapter. It's only made up of three sections. And the sections, they're really short sections as well. So today's lecture is going to focus on section 13.1, identifying the substance of the gene. And this kind of talks about how DNA was discovered to be a transforming principle, passing on genetic information to offsprings, and then I'm going to talk a little bit about some of the experiments behind that. So today's objectives: identify clues that bacterial transformation yielded about the gene, and how did bacteria viruses help us identify the genetic material, and then describe the role of DNA in heredity, basically the three roles of DNA in heredity. So we have to talk about a man named Avery Griffith, who was doing experiments, I believe, in the 1920s. I might be off on that by a couple of decades, but he was studying pneumonia, basically the bacteria that causes pneumonia, and he found that there was some type of transforming principle in the strands of bacterium. So what I mean by that is he was studying two strands, okay, a disease-causing bacteria, okay, the one that actually causes the illness and usually resulted in death. And then he was studying another strand of the same species, but it was the harmless version. There was a mutated form, and it was the harmless version. So you'll see here it says S-type. Well, he noticed that the disease-causing bacteria, when it was cultured into a petri dish, this is a petri dish with some agar, the edges of the colonies were smooth. So that's what S stands for, smooth. And the harmless colonies were rough and bumpy. So his experiment was pretty simple, okay? He took disease-causing bacteria and infected mice with it, and the mice, the mouse died of pneumonia. He took the harmless bacteria, injected it into the mouse, and the mouse lived. Now, bacteria are usually, they can be killed in a variety of different ways, and one of them is temperature. So if you raise the temperature high enough, the bacteria will, die, and we use this for sanitation purposes, today. So what Griffith did is he killed the disease-causing bacteria. So he killed it, injected it into the mouse, and the mouse lived. So then Avery Griffith did one more step, a brilliant step. He said, if I take the heat-killed disease-causing bacteria and I mix it with the harmless bacteria. So notice the outcomes for both of these separate. Heat-killed disease-causing, the mouse lived, harmless bacteria, the mouse lived. So he's like, well, I predict that the mouse should live. So he took the heat-killed disease, mixed it with the harmless, injected it into the mouse, and all of a sudden, the mouse died. And this shocked him. He was like, wait a minute. How did this happen? And when he cultured it, he noticed that they were disease-causing again, like they had come back. So we call this transformation, when one type of bacteria had been changed permanently into another. So the harmless bacteria that was alive had somehow transformed into the disease-causing bacteria. So when things die, or are damaged, they tend to release things inside their cells. When you get a bruise, cells are released, they're damaged, right? They're busted open and they're spewing everywhere. And this is why we get bruises and they go through different colorations, because your body is like trying to clean up the mess. Well, when these disease-causing bacteria were heat killed, everything was released. DNA, organelles, just cytoplasm. And somehow that harmless bacteria took up something, from the disease-causing, and then transformed into disease-causing. So harmless converted into disease-causing. So the ability to cause disease was inherited by the offspring. So Griffith concluded that, hey, that transforming factor has to be a gene. So he did more experiments, to try to identify DNA as the transforming principle. And one way he did that was through a chemical analysis. So DNA, the known value of DNA, we basically study the percentage of nitrogen to the percentage of phosphorus. And we're going to learn in the next section that DNA is basically made up of nitrogen, bases, and a sugar phosphate backbone. And so the ratio of nitrogen to phosphorus is around 1.69. So when he analyzed different samples of the mice that underwent transformation, he got these values, and you can see that all of these values are really close to the known value of DNA, but it still wasn't accepted by the scientific community. So he did some more experiments. He said, okay, did transformation still occur even though I didn't inject it into the mouse? Maybe we tried a different organism. Maybe we just analyzed it in the petri dish. Transformation occurred. Okay, so the mouse had nothing to do with this special ability. What if I removed a sugar coat? So I'm guessing they're removing, I don't know what the sugar coat is. The sugar coat of a bacteria cell wall, maybe, I don't know. But transformation still occurred. What if I removed the proteins? Maybe we add some enzymes that digest proteins. Did transformation still occur? Yes, it did. So there's no way protein is this transforming principle. What if we got rid of the RNA? Now, I know you probably don't know what RNA is because we haven't discussed it yet, but we're going to talk about the DNA dogma, how information flows from DNA to this intermediate molecule called RNA, and then it's going to be converted into proteins, and proteins is what is responsible for carrying out chemical reactions inside of you and for some of the physical characteristics that you have, like pigmentation of skin and eye color and hair color and so on and so forth. And so when he added enzymes or some type of chemical that destroyed RNA, did transformation still occur? Yes. So he said, all right, what if I added some enzymes that broke down DNA? Did transformation still occur? No. So he's like, okay, so it's got to be DNA. But you know what? Scientists were still skeptical of Avery's work because they knew that chromosomes contained more protein than DNA. So in a previous chapter, we talked about what chromosomes are made up of. DNA wrapped around these proteins called histones, and you form a nucleosome, and it gets super tightly wound, compacted into those X-shaped chromosomes that we know. So it wasn't until Hershey and Chase, a set of different scientists that were studying a virus called the bacteriophage. This virus is very specific to bacteria. Okay, it only affects bacteria, and they were using this virus to study if maybe DNA was that DNA transforming principle. So how it works. Viruses, what they do is that they infect host cells, and they inject their DNA into a host cell, and then that host cell takes in that DNA and starts to make more of that virus. And so that's basically a virus in a nutshell. A virus is not considered a living thing. Okay, it's either active or inactive, or latent or lytic. And what was I going to say with this? It's not alive. It cannot copy itself. It cannot respond to its environment. It can't do some of the characteristics that are qualified in order to be a living thing. Viruses are excellent in this study because viruses are only made up of two things. Okay, they are made up of a protein coat, and inside that protein coat is DNA. So just a great way to study whether or not DNA is that transforming principle. So Hershey and Chase confirmed that DNA was the transforming principle, and I'm going to describe that onto the next slide. They discovered which part of the phage, either the DNA that's inside the protein or the protein coat, was injected into bacteria. So how they did this is they took two different types of bacteriophage. Okay, so they're the same species, but this bacteriophage was labeled. They labeled the DNA. They tagged the DNA with phosphorus 32. Now you're probably wondering, what does that mean? Well, believe it or not, we can tag molecules and follow their fate inside of cells, usually with some type of radioactive isotope. Okay, and so this bacteriophage, all we did was we marked or tagged the DNA, and we are going to follow the fate of DNA. A different type of bacteriophage, we tagged the protein coat and left the DNA untagged. So we tagged it with sulfur 35 in the protein coat. So we let the phage do its thing. Okay, the phage infected the bacterium, and then we cultured it, and then we looked to see where the radioactivity was found. If it was found inside the cell, then and if it was phosphorus 32, then that would indicate that DNA got injected into the host cell, and then the host cell started to read those genes and then make more viruses. If it lit up, like outside the cell here, That means that protein doesn't enter the host cell and doesn't transfer that information. So protein is not the transformative principle. DNA is. Okay, so there was no radioactivity inside the bacterium. So thus it concluded, hey, DNA is the transforming principle. Okay, now the role of DNA, there's three basic roles. They store genetic information. They copy that information during the process of replication, especially in interphase during the S stage. So that way, when it goes through cell division, the new cell has the same code of instructions. And then gene expression puts the genetic information to work by making proteins. So yeah, that's the three roles of DNA. All right, so that does it for 13.1, identifying the substance of the gene.